In this video, we will be covering the musculoskeletal system. The musculoskeletal system is composed of the bones, joints, muscles, tendons, ligaments and bursae of the body. The musculoskeletal system gives shape and support to the body and enables the body to move. The skeletal system provides the body's framework as well as its foundation. The skeleton provides support and movement, and it is achieved by the combined action of muscles, tendons and ligaments attached to the bones. First of all, the interaction between bones and muscles drives a multitude of movements. Also, many muscles, arteries, veins, nerves derive their names from nearby bones. For example, radial artery. Finally, bones provide ready landmarks as clinicians navigate their way around the human body. For all of these reasons, learning the names of the body's major bones is a key part of understanding human anatomy and physiology. The major functions of this system are to support and protect the body and foster movement of the extremities. The musculoskeletal system provides protection for vital organs, including the brain, heart, and lungs, serves as a framework to support body structure and makes mobility possible. Muscles and tendons hold the bones together and joints allow the body to move. They also move to produce heat that helps maintain body temperature. Movement facilitates the return of deoxygenated blood to the right side of the heart by massaging the venous vasculature. The musculoskeletal system serves as a reservoir for immature blood cells and essential minerals, including calcium, phosphorus, magnesium, and fluoride. Calcium and other minerals are stored in bone tissue. More than 98% of total body calcium is present in bone. Many components of blood are made in bone marrow. Bone is a strong and durable type of connective tissue. It consists of water, 25% of water, organic constituents including osteoid, the carbon containing part of the matrix, and bone cells, that is 25%. Inorganic constituents, mainly calcium phosphate, that is 50%. Although the bones are hard and rigid but are living structures built and maintained by living cells. If damaged, they can bleed and are painful, and if broken, they can mend. Continuous activity takes place inside the bones as they carry out their functions. Now we will see the functions of the skeleton or bones. It supports the body, provides the framework of the body using bones of various shapes. It gives shape to the body. It provides attachment for muscles and tendons. It permits movement of the body as a whole and of parts of the body by forming joints that are moved by muscles. It protects the internal organs. It forms the boundaries of the cranial, thoracic and pelvic cavities, protecting the organs they contain. The skull protects the brain, the spine protects the spinal cord, the rib cage protects the heart and lungs, and the pelvic protects intestine, urinary bladder and female reproductive system. It produces blood cells. All the red blood cells, white blood cells and platelets in the blood are made in the red bone marrow, and it contains red bone marrow in which blood cells develop, that is called hematopoiesis. And also it stores calcium and other minerals so that there is always a supply readily available for release when needed by the cells. It provides a reservoir of minerals, especially calcium phosphate. Now we will see the structure of bones. The bones are considered organs because they contain various types of tissue. They are not only dominated by osseous, that is bony tissue, but also contain nervous tissue, cartilage, fibrous connective tissue and both muscle and epithelial tissues. Nervous tissue is found in bone nerves, whereas cartilage is found in articular cartilages. Fibrous connective tissue lines bone cavities, and both muscles and epithelial tissues are found in the blood vessels of bones. The external layer of bone is called compact bone, which contains spongy bone that is made up of small, flat or needle-like pieces called trabeculae. It appears smooth and solid when we see. Open spaces between trabeculae are filled with red and yellow bone marrow. Spongy bone is made up of open struts and plates covered by a thin cortex of compact bone. This covering is also referred to as cortical bone. In bones, struts can refer to either the trabeculae that form the internal or honeycomb-like structure of cancellous bone, or to grafted bone pieces used surgically to reinforce weak or fractured areas. Bones are described as short, irregular or flat, are all made up of thin plates of spongy bone covered by a combat bone. The plates are covered by connective tissue membranes, that is the periosteum outside and the endosteum inside. These bones have no shaft or epiphysis because they are not cylindrical. Inside they have bone marrow between their trabeculae, although there is not a well-formed marrow cavity. Hyaline cartilage covers the surfaces of these bones where they form movable joints with the nearby bones. The spongy bone in flat bone is called the diploid. Nearly all long bones have the structure of a shaft, bone ends and membranes. The shaft is known as diaphysis. It is tubular structure forming the long bone axis made up of thick collar of a combat bone surrounding a central medullary cavity. This cavity is called the yellow marrow cavity in adults because it contains fat that is called yellow marrow. The bone ends are called epiphyses, which are usually broader than the diaphysis. Inside they have spongy bone, whereas their outer shell is made up of combat bone. The joint surface of each epiphysis is covered by a thin layer of hyaline cartilage. This layer cushions opposing bone ends as they move and absorb stress. Between the diaphysis and each epiphysis of long bone is an epiphyseal line, which is a leftover remnant of the epiphyseal plate. There is also a disc of hyaline cartilage that develops during childhood, lengthening the bone. At the point where the diaphysis and epiphysis meet together is a flared portion that is sometimes referred to as the metaphysis. The external surface of a bone, except for the joint surfaces, is covered by a shiny, white and double-layered periosteum, a membrane that is richly supplied with blood vessels and nerve fibers. Dense, irregular connective tissue makes up the outer fibrous layer. Inside, an osteogenic layer touches the bone surface and is made up of mostly of primitive stem cells known as osteogenic cells. These cells form all bone cells except for those that function in bone destruction. The bone vessels and nerve fibers of the periosteum pass through the bone shaft, entering the marrow cavity through openings known as nutrient foramina. Groups of collagen fibers called perforating fibers or Sharpie's fibers extend from the fibrous layer into the bone matrix and bind the periosteum to the underlying bone. The periosteum also serves to anchor tendons and ligaments in areas that are extremely dense. Metaphyseal vessels supply blood to the diaphyseal surface of the epiphyseal cartilages, where they are replacing bone. Periosteal vessels provide blood to the superficial osteons of the bone shafts. During endochondral bone formation, they branch to enter the epiphysis and provide blood to the secondary ossification centers. The endosteum is a delicate connective tissue membrane covering internal bone surfaces. In most trabecular cavities of spongy bone in long bones and in the debris of flat bones are found hematopoietic red marrow. As a result, both these cavities are referred to as red marrow cavities. The endosteum covers the trabeculae of a spongy bone and lines canals that pass through combat bones. The endosteum also contains osteogenic cells, which may differentiate into various bone cells. In the newborn, red bone marrow is found in the medullary cavity of the diaphysis as well as all areas of spongy bones. In adults, most long bones have a medullary cavity that contains fat which extends a good length into the epiphysis. Very little red bone marrow is found in an adult's spongy bone cavities. As a result, red blood cell production in adult long bones usually occurs only in the heads of the femur and humerus. There is a greater amount of hematopoietic activity in the red marrow of debris of flat bones such as the sternum and certain irregular bones such as the hip bones. A yellow marrow in the medullary cavity can convert back to red marrow if there is significant anemia requiring more red blood cells. We will see bone cells. There are 5 major types of bone cells. They are osteogenic cells, osteoblasts, osteocytes, bone lining cells and osteoclasts. All except osteoblasts originate from mesenchymal cells. Each cell is basically a specialized form of a certain cell type that becomes mature or functions in a certain process involving bone growth. Like various connective tissue cells, bone cells are also surrounded by their own self-made extracellular matrix. Now we will see in detail. Osteogenic cells, also known as osteoprogenitor cells. These mitotically active stem cells are found in the periosteum and endosteum. They are squamous or flattened cells when bones are growing. Stimulation of these cells causes them to often differentiate into osteoblasts or bone lining cells. These cells help differentiate osteoblasts and bone lining cells. Others may remain as osteogenic cells. Osteoblasts are the cells responsible for building new bone tissue. These are derived from cells thought to be associated with blood vessels. Osteoblasts' function in bone formation is to secrete bone matrix. They create and release a mix of proteins called bone matrix. Bone matrix is made up of proteins like collagen mixed with calcium, phosphate and other minerals. These cells are related to osteoprogenitor cells, osteocytes, fibroblasts and chondroblasts. Osteoblasts have three main functions: growing new bones, that is bone formation, reshaping bones to help them change as the age grows, healing damaged or broken bones. Osteoblasts are triggered by chemical reactions or hormones when a bone grows or changes. After they are activated, osteoblasts move into place and deposit bone matrix in spaces on a bone that needs to grow or strengthened or repaired. After it is in place, the bone matrix solidifies and hardens into new healthy bone. Once the osteoblasts have completed their job of laying down new bone tissue, they can then become part of the bone by transforming into osteocytes, or they die if they are no longer needed. Osteoblasts are the bone forming cells that secrete collagen and other constituents of bone tissue. They are present in the deeper layer of periosteum, and they are present in the center of ossification of immature bone, and they are present at the ends of the diaphyses adjacent to the epiphyseal cartilages of long bones, and they are present at the site of a fracture. As bone develops, osteoblasts become trapped and remain isolated in lacunae. They stop forming new bone at this stage and are called osteocytes. These are mature osteoblasts that have become embedded in the bone matrix. Osteocytes are nourished by tissue fluid in the canaliculae that radiate from the Haversian canals. Their functions are not clear, but they may be associated with the movement of calcium between the bones and the blood. Osteocytes are mature bone cells involved in bone maintenance. They occupy small cavities called lacunae in the bone and have protoplasmic projections connected with the same structure of other osteocytes. The osteocytes conform to the shapes of the lacunae. These connections forms a system of tiny canals within the bone matrix and act to maintain it as needed. When osteocytes die, the matrix surrounding them is resolved. They also react to strain or stress and respond to stimuli such as bone deformation, bone load and weightlessness. The osteocytes allow the osteoblasts and osteoclasts to build up or degrade the bone matrix as needed. This preserves calcium homeostasis. Osteocytes are distributed within the lamellae, forming the network that maintains the viability and the structural integrity of bone. The Haversian canal is located in the center of the osteons, containing blood vessels and nerves. Blood vessels facilitate the exchange between osteocytes and blood. The vascular network provides structural support, nutrition and a waste removal system within this space. Bone lining cells are flat cells on bone surfaces where bone remodeling does not occur and are believed to also help maintain the bone matrix. On external bone surfaces, they are called periosteal cells, and when they line internal surfaces, they are called endosteal cells. Next cell is called osteoclast. Osteoclasts are located in shallow house-shaped lacunae, that is small pits in bones. They are multinuclear cells involved in dissolving and resorbing bone to maintain the marrow cavity. Osteoclasts are large multinucleated bone cells found at the site of bone resorption, which is called osteolysis. Osteolysis is a progressive condition where bone tissue is destroyed. They form from the hematopoietic stem cells that also differentiate into macrophages. The microscopic function unit of mature cortical bone is the osteon or Haversian system. The center of the osteum is a Hawaiian canal, contains a capillary. Around the capillary are circles of a mineralized bone matrix called lamellae. Within the lamellae are lacunae that contain osteocytes. These are nourished through tiny structures called canaliculi, which communicate with adjacent blood vessels within the Hawaiian system. Lacunae and calicellous bone are laid in an irregular lattice network known as trabeculae. Red bone marrow fills the lattice network. Capillaries nourish the osteocytes located in the lacunae. Osteoclasts dissolve and break down old or damaged bone cells. They make space for osteoblasts to create new bone tissue in areas that are growing or need repair. If osteoblasts are built-based, osteoclasts are bone demolition crew. Osteoclasts release enzymes that break down old bone. Osteoclasts are also known as osteophages. They secrete an acid that dissolves the matrix. They trigger chemical reaction on the surface of old bone tissue that dissolves it and creates a space for newer, stronger tissue to form in its place. The enzyme osteoclast release breaks down hardened bone matrix and reabsorbs it into the body. This leaves microscopic pits and divots on the surface of the bone. Once the targeted tissue in those places is dissolved, osteoblasts move in and deposit new bone in the same spot. They resorb bone matrix throughout life, replacing it with osteoblast. The process of breaking down areas of old tissue is tightly regulated or controlled and specific. Osteoclasts only target specific areas that have been tagged by osteocytes. These opposing processes, resorption and deposition, are regulated by hormones that control blood calcium. Osteoclasts have an important role to play in liberating minerals and other molecules stored within the bone matrix. Bone tissue serves as an important source of essential minerals including calcium and phosphate and other biological molecules such as growth factors. When calcium is released from the bone, this plays a role in maintaining homeostasis. Osteoclasts are regulated by different signaling pathways and molecules. Increased osteoclast activity leads to increased resorption of bone. During fractures and bone healing and certain disease conditions, osteoclasts use enzyme to excavate passages through the surrounding tissue, breaking down the calcified extracellular matrix. And at this point, they have an irregular border that contacts bone directly. This border has deep plasma membrane foldings that greatly increase the surface area for bone degradation via enzyme activity. The infoldings close off the surface area from the matrix surrounding it. The function of osteoclast is resorption of a bone to maintain the optimum shape. This takes place at bone surfaces under the periosteum to maintain the shape of bones during growth and to remove excess callus formed during healing of fractures, and around the walls of a medullary canal during growth and to canalize callus during healing. A fine balance of osteoblast and osteoclast activity maintain normal bone structure and functions. The cells responsible for bone formation are osteoblasts. Osteoblasts and chondrocytes develop from the same parent fibrous tissue cells. Differentiation into osteogenic cells rather than chondroblasts is believed to depend upon an adequate oxygen supply. This may be a factor affecting healing of fractures. That is, if the oxygen supply is not enough, there may be preponderance of chondroblasts, resulting in the cartilaginous union of the fracture. Cartilage functions at lower oxygen tension than most other tissues. Chondrocytes are a specialized cell that are the only cells found in cartilages responsible for producing and maintaining the cartilage's extracellular matrix. They are vital for the structure, support, and cushioning functions of cartilage in joints and other areas of the body, and they play a role in bone development and fracture repair. Covering the bone is a dense fibrous membrane known as the periosteum. This membranous structure nourishes bone and facilitates its growth. The periosteum contains nerves, blood vessels, and lymphatics. It also provides for the attachment of tendons and ligaments. The endosteum is a thin vascular membrane that covers the marrow cavity of long bones and the spaces in cancellous bone. Bone marrow is a vascular tissue located in the medullary cavity of long bones and in flat bones. Red bone marrow located mainly in the sternum, ilium, vertebrae, and ribs in adults, and is responsible for producing red blood cells, white blood cells, and platelets through a process called hematopoiesis. In adults, the long bone is filled with fatty yellow marrow. Bone tissue is well vascularized. Cancellous bone receives a rich blood supply through metaphyseal and epiphyseal vessels. Periosteal vessels carry blood to combat bone through minute Volkmann canals. In addition, nutrient arteries penetrate the periosteum and enter the medullary cavity through foraminae, that is, small openings. Arteries supply blood to the marrow and bone. The venous system may accompany arteries or may exit independently.